Recording. Okay, hello everyone. Welcome to MACSMIN, Mathematics and Computer Science for Materials Innovation. It's a great pleasure to introduce our first speaker in 2023, Professor Elspeth Garman from the University of Oxford, who will be talking about quantifying and comparing radiation damage in the protein data bank. Over to you, Elspeth.
Thank you. Yeah, good. Okay, just checking it works. Okay, thanks very much for the invitation to come to Liverpool. Very nice. I met a dead rat as the first thing that I saw when I came out of the station. And I thought, but apart from that, it's been all positive. Yeah, I'm going to talk about looking for radiation damage in the in 94,000 structures in the protein data bank of proteins. But I'm going to sort of start from the bottom up to try to explain why this is an issue and why we've done this work. I'm slightly echoing. I hope you can hear me, Dan. Okay, can you hear me?
Yes, Elspeth.
Yeah.
Is it convenient? Yeah, maybe you're closer to that corner.
This corner. Okay.
Yeah. So let's see the main video from the room.
Yeah. So the closest people look very, very large is the problem. So, right. So what I'm going to cover is what the problem is that we were trying to tackle and reveal, as it were. Why it matters to protein crystallographers, this problem of radiation damage in the structures in the data bank, and whether we can detect it in a single isolated protein data bank entry, rather than in a radiation damage series, of which there are only 23 in the very large database of protein structures, and then show you some results. So first of all, because when I was a nuclear physicist, when I was trained, I didn't know what a protein was when I changed fields in when I was 34, I thought I'd just run through, because if you don't know what a protein is, then none of this makes any sense at all. So proteins are long chains of 21 amino acids found in the body, and we can very, very easily, the chemists can sequence them trivially now and tell us which amino acid of the 20 is in each place. So we get something like this, which is like a chain of beads. But what we really want to know is how it's folded up like wet spaghetti in a sort of ball, because we want to know which of these amino acids are on the outside of the molecule, which are on the inside, which are in the active site. So these amino acids are all actually trivially the same in terms of, you see, they've all got the same CO2 and nitrogen at the top there. And then if we go back, we can see that they've all got different R groups on them, and they form together in, I hope you're familiar with the colors. Oxygen's red, nitrogen's blue for some reason that I've never discovered, and sulfur is yellow, and the carbons are white or gray, and the hydrogens are not shown. So they join together in these long strings by a reaction giving off water. And so the repeating unit is NCC, NCC along the string of the beads. And then the actual beads themselves are the different R groups that are attached to the amino acids that identify them. And these R groups can be just hydrogen, which is glycine, and then all these different things. And two of them have sulfur in. And how do they wrap up like wet spaghetti? Well, protein crystallographers, rather irritatingly but usefully, usually show the string that goes through the beads rather than the beads themselves. So this is the string in human necrosis factor, and you can see that even in your wildest, maddest AlphaFold 2 dreams, you wouldn't dream that it would fold up like this. There are millions and millions of possible conformations of this string of beads. If we show what it would really look like, if we could see it under a microscope, you can see you can tell nothing from this, because you can't tell where the conductivity of the amino acids is. You can't see where the string's going through it. And sometimes we show them as ribbon diagrams, and as well like this, with these zinc ions in this particular one. And you can see that they get quite complicated. This is one that I solved with great pain and anguish from cholera toxin. So what's the use of these? Well, if we have the structure of penicillin, for instance, then we can try and design better or more effective antibiotics because of antibiotic resistance. Classically, insulin, which is what I always use when I'm trying to tell a taxi driver or someone I meet on the train what I do. I say, have you got a friend who's a diabetic? They always know somebody who's an injecting diabetic. And I say, oh, well, because of knowing the three-dimensional structure of insulin, from Dorothy Hodgkin's 30 years of effort, we know what beads are on the outside of the protein, what are on the inside of the protein, and we can modify them to be more slowly absorbed by diabetics so your friend doesn't have to inject so often. That's why I'm a protein crystallographer. And in 30 seconds, you've got it right. And they get it because you need to know the shape, the three-dimensional shape. We have a great history of building up the size of the molecules, which we can solve by protein crystallography, from penicillin, which was 25 non-hydrogen atoms in 1949, and vitamin B12, both Dorothy Hodgkin got the Nobel Prize for these two structures in 1964, and insulin, which after, as I say, 30 years of work, she solved, which was 829 non-hydrogen atoms, and that was 1969. In 2009, Venki Ramakrishnan and Tom Stites and Adi Yonap got the Nobel Prize for solving the structure of the ribosome, which is the machine that does it's part of the one-way system of the central dogma of molecular biology, which is your DNA going to RNA being translated into protein. And the only thing that can go backwards along there are retrovirus such as HIV. So this one has 293,658 non-hydrogen atoms, just to give you an idea of the size of the problem that we can now address with protein crystallography. So if you haven't ever met the Protein Data Bank, I'm sure most of you have probably heard of it, but it was the first big data repository basically formed. It started in 1976. And what we have to deposit before we can publish our structures is the XYZ coordinate in three-dimensional space of the non-hydrogen atoms, and then a disorder parameter called the temperature factor or bin factor, which basically is an idea of the locus of movement of that atom. So if we can, whether it's just like this, or whether it can move either isotropically in a sphere, which is just one parameter we have to fit, or anisotropically, which unfortunately is 9 parameters. So you've got to have very highly detailed data in order to fit an anisotropic B factor. And then an occupancy, which is usually 1. So this just shows the increase in structures in the Protein Data Bank since it started. And last night there were of X-ray structures of 175,119, but the 200,000th structure has just been deposited about 2 weeks ago in the Protein Data Bank, because now we have NMR structures and an increasing number of electron microscopy structures. So it's grown hugely because of improving technology. Detectors, molecular biology has contributed enormously to that, because you can now make large volumes of protein, get E. coli to do it for you rather than mashing up old livers and hearts from their abattoirs. We had to do when I started the lab van, used to go and bring this tray of raw stuff back from the abattoir, and then you had to get the protein out of it. Structural genomics projects now out of fashion, but have hugely contributed as well to this. So this has just stopped working, so it's probably had too much rest because I'm talking too much. There we go. So how does this work? What's protein crystallography involved? First of all, you've got to get pure protein, which is, as I say, E. coli's job. Then you've got to grow a crystal, and then you've got to collect data, then you've got to do the structure determination, and then you've got to deposit the atomic coordinates, which is what's in the Protein Data Bank. So what is a protein crystal? And this is where the radiation damaged part starts to come in, because unlike small molecule crystals, which are used for drug development, for instance, they have a lot of solvent involved, because the protein molecules, as you've seen from that blobby mass, is not going to pack with no space between to make a crystal. So what's in between is a solvent, the magic solvents we try and grow the crystals from, the buffers, crystallization cocktails, and mainly water. And you can imagine the protein crystal being like synchronized swimmers in a swimming pool. And it's this water which gives us the real trouble, because when X-rays hit water, we get radiolysis, we get products, we get radical production, and that then gives us the radiation damage to the actual protein molecule as well. And Kathleen Lonsdale had a wonderful quote where she was trying to say, if you look at these ladies you see that they've all got different length noses. They've got their fingers in slightly different places because all those protein molecules aren't actually identical. There's dynamics involved. So a loop might be like this or like this. And what we get from our experiments is the average of the time and the space average of millions and millions of molecules. But she said a crystal is like a class of children arranged for drill, but standing at ease, so that the whole class as a whole has regularity, both in time and space, but each individual child is a little bit fidgety. And I really like that quote. So I'm going to move swiftly on. There's a black magic, and I could talk all day about how hard it is to grow crystals and how some people can do it, some people can't. Move on to the data collection. How do we actually collect the data? Well, the basic setup is that we have a free electron laser, a synchrotron, or a lab X-ray source. We have some optics to condition that beam. We don't really have a lens that works for X-rays, so that has to be quite clever. We have a crystal, which here is totally out of proportion, as you can see, because normally we can now deal with 5 micron size crystals. So, and at home, we can deal with one that's a 20th of a millimeter in size. So these are really small. You have to deal with them under the microscope. What's more, you haven't got to let them up, go bare in air because they dehydrate and then the order disintegrates. Then we've got fantastic detectors now, very large, very fast, and very single photon accurate. So very, very low background detectors. And then the goniometer is because I want to sample this protein molecule in 3 dimensions. So I've got to be able to rotate the crystal around and very accurately. And now the detectors are so fast that what I can do is put my X-ray beam on and just go like this all the way around without stopping and starting. So I don't rely on a shutter stopping and starting, and this has improved the accuracy of our data hugely, because you used to have to correlate the movement with the motor with the opening and closing shutter, and that used to give a lot of technical problems. Okay, so what do we do? Well, here is a crystal. It is cryocooled at 100 Kelvin, where we get about 70 times more data than we do at room temperature because of radiation damage. Take it to a synchrotron. Here is diamond light source. We get beautiful diffraction if we're lucky bunnies. And then we use Bragg's law, which I'm going to remind you of in a minute, just in case your O-level physics is, or your GCSE physics is gone. And we do the magic of the Fourier transform, which I'm not going to talk about today. And we get some electron density, which is this green chicken netting. And then what we have to do is fit what we know the sequence is to that. And that's the process of refinement. And if you're lucky, you get, that's the best one I've ever got, because you believe in atoms. So for proteins, we normally see sausages like this between the atoms rather than single atoms. But that was a very large enzyme that I got fantastic crystals of. And then it's quoted in the Melbourne Age newspaper as having seen balls rather than sausages. Very embarrassing. But anyway, what happens when we put the X-ray beam at room temperature on our crystals is really quite spectacular. So these are still in the tray in which they were grown, which can be put up on the I-24 microbeam line at Diamond Light Source, run by Robin Owen, one of my ex-graduate students. And the crystal here, a bovine enterovirus, is about 50 microns across, and you take 2 images, and then as you move the tray, and as the bit smashes up because of the radiation damage, you take 2 more images from the next bit, and then you move it again. And through doing this from about, I don't know how many crystals they used, about 50, they were able to get a complete data set. And just to show you what the beam can do to crystals, they rather pseudily etched their beamline number onto this crystal. So the X-ray are doing something. Now, what happens is that the lovely diffraction pattern, where this is the high resolution data, this is the low resolution data, here's Bragg's law. So the bigger theta is, lambda is fixed, it's the incident X-ray energy, the D is going to get smaller if theta gets bigger. So the big angle stuff is the small distances between the planes that are diffracting, or the waves are interfering. And as you irradiate it, what you see is that the detailed stuff starts to completely disappear. So we can't see any interference patterns from the side chains because they're all being zapped off by the X-rays, or rather they're being disordered rather than cleaved off. Sorry, they're being disordered. So the crystal is becoming the children about to go for lunch, basically, instead of all lined up for drill. So if we cool the crystals to around 100K, what we manage to do is these pink guys are the secondaries. So we get an initial absorption event, either on the protein, a direct event, or on the solvent, an indirect event. And then that produces one absorbed photon can produce up to 500 ionizations. So there's enough energy at 12.4 keV incident energy to ionize 500, a maximum of 500 other atoms. So you can see that there's a problem here. The pink guys, we thought we'd immobilize the whole lot to 100 Kelvin and they just sort of stay there. So that's why you get more lifetime, because although you can't do anything about the red ones, the primary events, because that's a fact of life, just like when you go for an X-ray and your bones absorb more than your flesh does and you get a negative image of yourself. The crystals, you can't stop them absorbing X-rays. That's going to happen if you put matter in front of an X-ray beam. So what we see is that at 100K, the intensity of the reflection stays high. It doesn't die off in the way I just showed you. And also we're able to collect higher resolution data, these angstroms here, not because it diffracts better at 100 Kelvin, but because it diffracts well for longer. So I've never found one that diffracted at 100K and it didn't diffract at room temperature. But if you ever hear of one, I'd love to know about it. So can we do anything about this? Well, everybody started cryocooling their crystals. Now you might say, well, I don't go around at 100 Kelvin. What use is it knowing protein structures of my body at 100 Kelvin when physiologically I'm at 37 centigrade? Well, the answer is that the main chain we know stays the same. Very, very, very largely, whereas some of the side chains will take up different conformations because you're taking out the dynamic disorder when you cool to 100 Kelvin. So the blue ones here are cryo. There's one at 0 Kelvin in the protein data bank, which worries me a little bit. You don't have to specify, unfortunately, with this. Some of them are not specified. But you can see that this is up until halfway through 2019. So spectacular cryo cooling has saved third generation synchrotrons, which were really far too, the flux density was far too high for protein crystallography because they just killed the crystals straight away. Now we have new things like serial crystallography, where you have a stream of crystals coming and you, if you're lucky, you hit one as the beam comes down. And that you can do room temperature experiments. So the interaction of X-rays with matter is what we're worrying about. So we like the fact they diffract, we want that bit, but as we know, it absorbs. This is Röntgen's wife's hand, which is the first ever X-ray, which I think was a bit mean. I think he should have done his own hand, but there we are. So what's happening here now is that we're getting diffraction, which we want, elastic Rayleigh scattering. But we're also getting Compton scattering, a low amount of that, but the photoelectric effect is what kills us. We get absorption, complete absorption of the X-ray, emission of a photoelectron at 12.4 keV minus the binding energy of that electron, which is tiny, and then we can spread the pain. So at 12.4 keV, which is 1 angstrom X-ray energy or wavelength, we get 8% elastic scattering, which is what we want, 8% Compton, 84% photoelectric absorption. So that's a rather nasty division. So we're getting 10 times the absorption as the elastic scattering, hence the problem. So how can we estimate what we're giving the crystal? We need to have some idea of the energy absorbed by the crystal. And the unit we use is rather inconvenient because it can only be estimated, it can't ever be measured, which is the dose. So dose is measured in the energy absorbed in joules over the unit mass in kilograms. And as we know, crystals, as I've said, are tiny and they're in nanograms or micrograms. And it's measured in gray, capital GY, and it takes care of the physics of what's happening, but not of the particular chemistry that I'll mention in a minute. You have to estimate it from the property of the beam and what's inside the crystal, because if it's got heavy things in, it'll absorb more X-rays than if it hasn't. So if it's got metals in, it'll have a shorter lifetime than if it hasn't got metals in. Now, just to give you an idea of the units here, here's a hamster. If I give it 3 joules per kilogram, it will go into total organ failure. I haven't done this experiment, I hasten to add, ever. But crystals seem to be able to tolerate 10 million times that. So 30 megagray is our absolute limit for biological crystals. And we reach that at a modern synchrotron in about 30 seconds of data collection. So if it takes you longer to take your data than 30 seconds, you've gone over the limit and you'll see damage to your crystal. So what sort of damage do we see? Well, as I say, we can plot dose along here. We have to be able to calculate it. My group have written a program over the last 10 years called RedDose 3D, which is open source on GitHub, which people use at all the nearly all the synchrotrons around the world now, which is great to try and find out how much time they have to take their data before they're going to damage the crystal. So we see the intensity dropping off even at 100 Kelvin as a function of dose here. And we have the dose to half intensity is a number that we tend to quote. But actually, you've zapped it far too much if you take it that far. So 0.7, taking it down to 0.7 is the 30 megagray limit. You see an increase in the unit cell volume. So this is a very strange thing. So hydrogen is being given off, but it's trapped in there. So the effect is that it collects at domain boundaries within the crystal, and then the unit cell appears to go up. This has been seen now in small molecule crystallography as well, but it's a very bad effect of actually irradiating the crystal. And then the scaling B factors, a data set will have a scaling B factor where you try to get it all on the same scale, because if you imagine if you hit me here, you're going to go through more of me and get more intense spots than if you hit me here, because there's not so much volume of me. So I need to scale within the data set as I rotate in order to get it all on the same scale. We see these going up linearly as well with dose. But more worryingly, and the reason we care about this, is that we get specific damage to particular amino acids. And the first thing to damage at cryo temperatures is disulfide bonds. This is lysozyme, of course, and there's 4 disulfide bonds in it. And in 3 of them, you can see where the sulfurs move to, which is the green density. The red density is, this is the 4th data set minus the 1st data set. So all these bits are exactly the same in both these data sets of contiguous data sets that have been collected. But here you can see that the disulfide bond is cleaved like this, and the green ones are where the new positions are. But if they've got a lot of space because it's outside the solvent, then they're just waving around like this, and you don't see the green density. You only see the fact that they're gone. So red is negative density, green is positive density. The next thing to go are, and you can just start seeing that on this one, are the disulfide bonds. Oh, first thing that happens, sorry, I should have said, is that all metallo centers are reduced. So we think that all the metallo enzymes in the PDB are actually in the reduced form. The next thing that happens is decarboxylation of asps and glues. And this happens in a very reproducible order in now hundreds of proteins, rather than sort of collateral damage over all the different amino acids. This is actually an oxidation reaction due to electrons popping, proton holes going the other way, electrons popping the other way. But it turns out that measured in 1987 was actually the fact that at 77 Kelvin, electrons can quantum mechanically tunnel at 77 Kelvin along the amino acid backbone as if it's a motorway, and find the most electroaphilic place in the protein, which is the disulfide bonds. So when that disulfide bond gets 2 electrons collected, it can scission. So that's what's happening. So I never thought that quantum mechanical tunneling would be at all relevant in my life when I studied it in physics, but here we have it causing trouble. If you've got any covalent bonds of heavier atoms, they rupture quite quickly. But the thing that this talk is about, and I'm at 35 minutes in, or rather 25 minutes in, is that the atomic B factors actually increase with dose. So we had X, Y, Z, which is the position of our atom in the protein data bank file. Then we had this B factor, which is the temperature factor, the locus of occupation of that atom. And then the occupancy was, as I said, is usually 1. And here, this is in angstroms squared, which is basically the area of the isotropic ball around the atom. And you can see it going up from 0 to 70 after a few data sets. So could we use this as a clue for an isolated protein structure in the data bank to say how damaged it is? Now, the problem is that we don't have more than one data set usually in the data bank. We've only got one. So we can't get anything from here because that's just where the atoms are. So the only thing we've got to play with is B. So in summary, what we see in radiation damage, in global radiation damage, which is in reciprocal space in the diffraction pattern, I've been through all these. We also see increased disorder, increase of the mosaic blocks. And then in the specific radiation damage in the actual amino acids, we also see the disulfide bond breakage. Here's the new positions of the sulfurs, the decarboxylation. We've actually detected a carbon dioxide molecule coming off in our electron density maps at 100 Kelvin. So this really happens, this gets chopped off. And then it was thought that the OH came off tyrosines, but we've showed a few years ago that this, the radiation chemists saying that this is impossible, it can't happen. And it turned out that it was a movement of the entire thing, which makes this look like it's come off, but it hasn't. Okay, so I talked a little bit about the radiation damage limit. You need to be able to calculate what the dose is, and that's for a dose 3D, which if you're interested, I can give you the link to. And we have this problem, which is that the specific damage happens way before the experimental dose limit of 30 megagrade, way before you start seeing your diffraction pattern shrinking. You're already damaging the amino acids. Now, why does that matter? Yes, that's right. Why does it matter? It matters because of three reasons. One is we don't get all the data we need because radiation damage is killing our diffraction. The second is that I've mentioned already three sorts of non-isomorphism, which is that this data set is not going to be the same as the next data set. The unit cell is increased. It turns out that it looks like the molecules actually moved within the unit cell in its position, and I've got damage. And if I've got a beam that is not a top hat, which very rarely are, they're usually Gaussian beams, then in the middle of the crystal, all the disulfides will be broken. Here, half of them will be broken, and here, none of them will be broken. So I'm going to end up with the average. Remember, I said what I'm going to see is the average of all the atoms, all the conformations. I'm going to get washed out electron density that's not as sharp as it could be, just because I'm in all these different conformations. But the main reason that we care about this is because it affects the biology, and that's what we're doing the whole thing for. So it's possible, that you're wrong about the state of your metal. For instance, photosystem 2 has got manganese in, which they've argued for years about what state it's in, whether it's got an electron or not. The X-ray beam is affecting those results. Decarboxylation of glutamate in some enzymes is part of the protein mechanism, but if they're all decarboxylated, how can you tell that that's the mechanism? And then bacteriorhodopsin is a classic case where a Japanese group has spent years untangling X-ray induced changes with real changes. So that's why we care. So, hey, we got there. Can we detect it in a single protein data entry? As I say, all we've got to play with is this B factor here. Can we give every PDB at 100K, I'll come why we can't do it at room temperature yet, a radiation damage index, so that the users of the protein data bank can see that they might be a bit suspicious. So one scary statistic is the number of millions of downloads per day that happen from the protein data bank compared to the number of protein crystallographers who know what the numbers mean. So they're treated as gospel, and we're having a problem at the moment in that people think the AlphaFold hypotheses structures are also gospel truth, and are using those because the sort of uncertainty index is less important than the actual AlphaFold 2. If you haven't heard of, has everyone heard of AlphaFold 2? Yeah, good. Okay, so can we do this? So I mentioned already that this B factor is, or the atomic displacement parameter is related to the locus of occupancy of the atom in its place. So it's basically its uncertainty of position or where you might find one with a probability. And we can do anisotropic. We've got really high resolution data, and small molecule crystallographers always do that because they always have enough measurements that they can do that. But it increases the number of parameters from 4, X, Y, Z, and the radius of this sphere to 9. So you've got to really have a lot more measurements to be able to do anisotropic. But the problem with this is that it's very related to the packing density. So we can't just say, oh, it's got a big B factor, it must have been damaged. Because it might be that it's on the outside of the protein, so it can move a lot anyway, which it will if it wants to, because it's only got one bond, covalent bond behind it. Whereas if it's in the middle of the protein, it'll be stuck like this. So you've got to somehow deconvolute the packing density from the actual B factor. And that's what we did several years ago with something called B damage metric. So this isn't the one we're finally going to get to, but it's a start. B damage. So the question is that glutamate actually moved so we don't see it anymore, or has it come off because we rarely see the carbon dioxide? So this would be radiation damage. That is mobility. So what is it that's happening here? So what we want to do is that in a high packing density environment, you know that it's not mobility because it can't move that far. It must have been damaged. So if you can take that B factor and remove from it the packing density element, you might be able to use that then for the radiation damage part. So this shows two residues in an enzyme that we studied in great detail. This one has a very low B factor in the refined structure, whereas this one has a high B factor. Now, that is because that one actually is at the surface of the protein and can move around a lot. This one's very internal. So its packing density, red is high here, is very, very high. And when I take that out, I find that what I'm left with, oops, gosh, I didn't want that. I wasn't expecting it to do that. I'm sorry. That was very good. Whereas this one is on the outside, it has a high B factor, but it has a very low packing density, and they end up with the same B damage parameter, even though they're in very different environments with very different starting B factors. So that illustrates that. So what did we do? Well, we calculated this index B damage by taking the B factor from the protein data bank and then looking at all the atoms in a similar packing density and taking the average of their B factor and dividing it out. Right, so the radius we used initially was 14 angstroms. So packing density within 14, we've now reduced that to 7 angstroms after testing many distances. Of course, the protein molecule will have a friend that's near that might affect the packing density. So we can't just take one molecule. We've got to take the way it's arranged in the unit cell, which is your favorite thing. So the trimmed unit density, we've got to consider any protein, other protein molecule that's in contact with it to get the packing density. So here you see the 7 angstrom radius we're taking. So this one has got more, it's got 6 in, that one's got 4 in, so that has a higher packing density than that one. So we take the average B factor from the protein data bank of this, and then that yellow atom would be the average B factor of window B here, that's window B, and that's window A, and so on. So is everybody happy with what we're calculating here? Any questions?
I have a question now.
Yeah.
Yeah. So do you just do it within a single entry, within a single protein, or you do it across all proteins?
You'll see in a minute, but for a single protein, B damage allows you to identify the damaged atoms, right?
Yeah.
Because it's high, but it doesn't allow you to compare it with another protein data bank entry, and that was our problem.
Right.
We got this far, and then we did the thing
I
that
see.
I'm going to tell you about next. Okay, so we tested this on a low and high dose, 6 pairs of data sets taken in identical conditions from the ESRF in Grenoble. And you see that this is the density of the B damage, sorry, down here it is, the B factor distribution of the entire structure, low and high dose. And you can see you can't really see the difference between this, and that this has a very small difference. This is B damage. This is of all of the atoms. This is of the cysteines, which are in the disulfide bonds, the aspen glues. So these are the ones that are affected most by radiation damage. And you can see there's quite a big difference on B damage, but not very much difference if we just take the B factor. So this is with the packing density deconvoluted. This is with just the raw stuff from the data bank. Now, this is just for the sulfurs. And you might say, well, why didn't you just use the sulfurs? The reason is that only 25% of the proteins in the data bank have disulfide bonds, which break. The other 75% they don't have them. So this is not a good thing to use on its own. So we have to do something different. So what we found was that if we plotted B damage against the loss in electron density and had a look at that for fairly damaged but not very damaged data sets, that we find a relationship between the electron density loss and the B damage. There's a lot of scatter on it. So this is for sulfurs. Oh, sorry. Sorry. Yeah, there we are. Don't touch. So this is for sulfurs, and this is for the acids and glue. So then we wrote a program which is in the CCP4 suite to calculate this, exactly the formulae that I went. And we then looked at 2,600 entries in the PDB, and we found that we set a filter on that, not room temperature, because they didn't obey anything. And we did a study of that to try and find out what factors were affecting radiation damage. And the only really strong result we had out was that there are 3 different conformations of disulfide bond, depending on how they're tethered by the amino acid, what the angle is of the 2 strings of the beads that are joining to make that disulfide bond, which is like a scaffolding bridge in the protein. And that it turned out that it was more easy to damage the staple one than this one, than these 2, because of the it's under more strain, basically because of the angles that we think. And then we were now able to give every single atom in a structure a number, which was the B damage number. You can see that that sulfur there is getting a bit damaged, whereas, and you can identify these in the protein here. And the Rabdown software to calculate this so that you can display your protein and find out the sites of damage is, as I say, available with radiation with CC4. And if you now look at the electron density in these places where the B damage was high, you can see that there's damage starting. So if we go back there, you see that was only 1.25, that one was 1.62, and you see the difference in the red electron density, which is one data set subtracted from another. So it's a high dose minus a low dose. So you can identify now where the problem is. But the problem is we can't compare. So a user comes along and wants a protein molecule from the protein data bank, and there's 2 entries. Which one should they choose? And there's no way to say, well, that one might be more damaged than that one. So what Catherine, my fantastic master's student, did, who's now with David Baker as a postdoc in Seattle, she tried, well, actually, my 6 ideas, none of them worked, to get one number out of the from this B damage metric, and then she had an idea and that worked. So that was brilliant, absolutely great. When you have a student that's far brighter than you, that's where you've made your career then. So what she tested, as I say, loads of things, but in the end, what she did was we had 23 damaged data sets from the protein data bank where people had deposited up to 7 structures taken from the same crystal. And by testing those to see what worked, what she ended up doing was taking the B damage values of the oxygens on the glutamates and aspartates, because as I say, a lot of them don't have sulfurs, disulfide bonds. And she plotted the low dose, which is the blue here, and the high dose B damage, and this is being kernel density smoothed. You can see the red plot down here. And then she took the median value of all of the B damage in the structure. The median, not the mean, because it turned out the mean could be really affected by some real outliers. So she took the median value and then took the low dose, no, don't do that, low dose over the high dose side. So the area, sorry, the high dose, other way around, the high dose over the low dose, the area of the right hand side of this over the area of the left hand side. So we've got the median of all the atoms, but the blue and red are only the oxygens from the glutamates and aspartates, which are the most sensitive to radiation damage. And amazingly, we got from the radiation damage series in the data bank, we got more or less the same slope with respect to dose, because they were all deposited with dose in the data bank. Now, you might say, well, I don't think they're the same. But remember that the dose has to be calculated from the parameters of the beam and at least a factor of 2 variation in how accurate that is. We think up to a factor of 2, because you've got to know the flux, the beam shape, the energy, which you know, you've got to know what's in your crystal, and you might have crystallized it in something with sodium chloride and not know the concentration, and that's got sodium and chlorine, which is more absorbing, and all of these things have to go into the calculation of dose, but they've been calculated with our software just because it's the only one available. Sorry, I'm not being big-headed here, it's my only way to do it. Okay, so that was, I said there were 23, and I'm just showing 7 of them there. I've got them all here, low dose, mid dose, and high dose. And what you can see is this Bnet number that we calculate here. If you take the asparagine and glutamines, which are not damaged preferentially, you get more or less straight lines with of the Bnet parameter from the data sets with those, whereas if you take the Bnet, it's definitely always going up. There are explanations for these two that don't seem to go up, but I won't go into those. So these are all the 23 series from the protein data bank. Now, if you just take the Wilson B factor, you can see that it's also slightly going up, but it's not a big enough signal figure to use compared to the Bnet. So what results did we get? Well, what's the filter? In order to calculate Bnet, we need people to have actually refined the atomic B factors. So the resolution has to be better than 3.5 angstroms. Otherwise, you don't usually have enough parameters, enough data beyond that at 4 angstroms to do individual B factor refinement. You tend to do entire side chains and say that's one block. The only protein structure so far, there's not enough data on nucleic acids in the data bank to do that. It doesn't work on room temperature data. Yes, I know I'm running out of time. Yeah, cryo data. And we also asked for more than 10 aspirin glue residues, because otherwise you get real outliers. There's not enough signal, basically. Resolution, as I said, has to be better than 3.5 angstroms. Now, it turned out that in November 2020, when we did our big screen of the protein data bank, 145,000 structures in it, and 93,938 to fulfill these requirements. That's why there's a little there. We were short of 13 to get 94,000. In fact, we had 94,000, but the reviewers asked us to reduce our resolution limit, so we lost a few. So what does this let us do? Well, blue is low, B damage, and red is high. So again, I can get for each atom, I can get a B damage, but now I've got one overall number for this structure, which is 1.4, which is good. Okay, as you'll see, that's a good number. So it's a derivative of B damage, this number Bnet. And just to give you an idea, 95% of our 93,000, sorry, it was 22 we were short of, not 13, structures has a Bnet less than 3. So we set the limit of where you should really look at the structure. If Bnet was bigger than 3, you really need to have a look to see what's going on. That what you see above 3 is definite asp glue damage on everything that has a bigger Bnet than 3. So you might say, but surely Bnet will be correlated with different parameters like the mass of the protein, the temperature of the data collection, how well it was refined, that the resolution, the number of aspirin glue residues, the B factor restraints used in the refinement. And we tested all of these variables, and this is the Spearman's rank correlation coefficient shown up here, and you see that indeed 3 of these parameters have huge correlation with Bnet. So that's not good. Okay, anything below 0.2 is fine, which all these. So we showed it wasn't correlated with the number of aspirin glues, the B factor restraint weights didn't affect it much, and the molecular mass. So it turned out that resolution is really, if you've got a high resolution structure, then you have a low R work and R free. Whereas if you've got a lower resolution structure, you tend to have higher. These are parameters, sorry if you're not familiar with them, as to how well your model fits the data. This is a test you do right at the end, the R3, where you take 10% or 5% of the data and you don't use it for the refinement, and then you put it in at the last minute, to see how well it agrees with your model, how well the model agrees with the data. Sorry, some of my students were saying they did the data to the model. No, the model to the data. Okay, so then we came up with a derivative of Bnet, which was a percentile, which was corrected for resolution, if you like. So you take the 1,000 structures from the protein data bank around that resolution and then normalize for that. And then we managed to deconvolute the R work and R free. So these are the percentiles of each structure and with the closest resolution. And now all of the Spearman, CC ranks are below 0.2. So then we've got the nasty ones. So I mentioned that 3 was a maximum. So here are 4 at 25, 42, 20, and 34. And these are the little diagrams you get if you've ever looked in the protein data bank with the quality. And what you see is that they all look okay. You look at those and say, oh yeah, they're blue ones. But then you look at the aspen glues and you can see, well, that disulfide there is totally broken. Don't touch. And the new sulfur positions you can see in green there because it's gone like this. And then the aspen glues are completely damaged. So we published the worst 10. So this is where everybody doesn't like it. The worst 10 in the whole database. There was one at 78, a Bnet of 78, but it only had 6 aspen glues, so we didn't put that one up because it was below our threshold of 10. This one, there's 2 here that don't make any sense at all. This one and this one. This one is just a really, really bad structure. I mean, you can't tell what the hell's going on there. But look at it. Well, it's really bad here, right? Most of the others look sort of okay, but this one's terrible. And then this one was a synthesized protein. In our first 10, before we did the resolution cut, there was actually another structure in there, which was looked a bit like this. But this was because it was a synthesized thing with just strings like this, and it had aspen glues at each end, and they were basically free to move around in the structure. I'm coming to the summary. So what can Bnet do? Well, it can detect potential sites of damage, rather can find the potential sites of specific damage, but you can't compare it easily between structures. Whereas Bnet is an overall number for one PDB, isolated PDB. You can calculate it with the CCP4 program. We're working very hard to get that into Phoenix at the moment, because once it's in Phoenix, the PDB say they'll consider adding it as a bar on the bottom of the thing. It's a long procedure to get the PDB to do something like that. But anyway, we're working on the first stage of that at the moment. But what worries us when looking at these structures is that at the moment, radiation damage is largely overlooked by users of the protein data bank. And many people don't look at electron density ever. They just look at the model and say, that's what it looks like. So Catherine Shelley is the star, but Marcus Gerstel did the work on B damage. He's was at Diamond Light Source, but now he's working for a cybersecurity firm. Johnny also worked on the software. He's now at Spotify. Tom was a master's student with me, and Charlie Burry is working for XAMP. I don't know where Tom, he was last singing in a choir, professional choir. I don't know where he is now. He also did a PhD in structural biology. And Catherine, as I say, is with David Baker, and I acknowledge the BBSRC and the Moritz Heinemann Scholarship who funded Catherine all the way through. I just wanted to end by saying that I found this quote, which you've seen already, but it's just brilliant. Because when I changed fields, I realized that on the very first day I had to ask if I didn't understand. Because I had a big argument with a student the very first day who came up from the microscope room and said, I've just seen a nucleus. And I'm like, no, I've been looking for a nuclei for 11 years in the nuclear physics department. I sure haven't seen one. And anyway, big argument. And he was very sticking to his guns. And eventually I realized, oh, he's talking about the nucleus of a cell. I'm talking about the nucleus of an atom. And I realized that the communication was absolutely vital, you know, and then unless I asked, I'd never know what was going on. And I asked, and some of the questions were really stupid in retrospect, but people would come up to me afterwards and say, oh, I've always wondered that, but I've never liked to ask. So most of the time, if you speak up, at least 30% of the people in the room will have the same question, weren't brave enough to ask it. This is from Nicola Fox, who's the new science chief of NASA. And she says there are no stupid questions. So please ask lots of questions. So thank you for your attention.
Thank you very much. Okay, so I'll stop the recording.
This is to say it's only going to get worse.